Hello class, so this is the second part of the series Nucleotide Biosynthesis. And we will be talking about key steps in nucleotide biosynthesis are regulated by feedback inhibition, and also disruptions in nucleotide metabolism can cause pathological conditions. And so, as far as the regulation of pyridine biosynthesis, what can be regulated is the enzyme aspartate transcarbamylase. And remember that as far as creating pyridine biosynthesis, you have the aspartate and the carbamoyl phosphate that ends up forming that pyridine ring structure that ends up being attached to a PRPP, ultimately making UMP, which goes to other steps in the reactions where you end up with your final product of CTP. So that CTP is used as an inhibitor of the enzyme aspartate transcarbamylase. That's why you can stop that production of that pyridine type of base. But as far as the purine type of base with ATP, actually that is used to stimulate that enzyme of aspartate transcarbamylase. So that's how one of the things to help balance the amount of pyridine bases versus purine bases for the nucleotides. And so, as far as the purine nucleotides being controlled by feedback inhibition, there are several sites that actually can be affected for stopping the production of certain types of purine nucleotides. So, as far as AMP, that can inhibit the enzyme that is used to convert IMP into AMP. And then also GMP can be used as an inhibitor for the enzyme that converts IMP into GMP. And if you recall in the second part of the nucleotide biosynthesis series, that actually GTP is used to create AMP, and ATP is used to create GMP. So that also helps out with this regulation of different types of purine nucleotides. But the purine nucleotides of IMP, AMP, and GMP can all be used as an inhibitor for stopping the commitment step of PRPP being converted into phosphoribosylamine, which we know this is the commitment step to create purine nucleotides. But these purine nucleotides, AMP, IMP, and GMP, also inhibits the ribose 5-phosphate to becoming that activated sugar of PRPP. So thus, when you have so many of IMP, AMP, and GMP that's around and blocking PRPP from changing into purine, then thus it has the direction of going and making pyrimidine nucleotides instead, or histidine. And so, as far as the regulation of the ribonucleotide reductase enzyme, this enzyme has two allosteric sites. One is the activity, and the other one is for specificity. And so, as far as the regulation of the overall activity, it gets inhibited by the deoxy version of ATP. And then from there, what actually stimulates it is actually ATP. So that is used to stimulate the ribonucleotide reductase. For the regulation of substrate specificity, if you recall from the last lecture, UDP will eventually make TTP, and also just remembering certain enzyme converting CDP into UDP, that's having TTP still as the final product. Thus, it is a inhibitor for production of um, DUDP, and also it inhibits the production of DCDP as well. And so, since we have enough TTP that's out, and we want to generate more of the other nucleotides, the TTP actually stimulates the production of DGDP, and actually it acts like a chain reaction to end up having uh, DGTP to stimulate more of DADP from being produced. And then if you have enough DATP, just know that, like as a reminder, that ribonucleotide reductase is inhibited, and if you have ATP, this will actually want to generate more of the TTP. So it's this chain type of reaction that is happening. Now, as far as the purines are catabolized for excretion, so nucleotides of cells undergoes continual turnovers, and this facilitates the proper balance between ribo and deoxyribo nucleotides, as well as the removal of damaged bases. And so, with this turnover, begins with the hydrolytic conversion of nucleotides to nucleosides by nucleotidases, which is shown here, where you have an example of AMP going through the hydrolysis with this nucleotidases, where that phosphorus group is left, gone, and so you have that nucleotide is now a nucleoside because there's no longer that phosphorus group. And then from there, as far as continual hydrolysis, removing removing and releasing ammonium. This is the adenosine deaminase, which ends up making this new nucleoside, and then going through phosphorylases with the enzyme nucleoside phosphorylases. It releases the sugar from there, and they're having the production of hypoxanthine. And this should look familiar because that was brought up in the second part of the series on the salvage pathways of purine, and this is actually used to help create the nucleotide IMP, which is this same structure, but this has the phosphate group that is attached. So then from here, you have oxidation and some hydrolysis with this enzyme xanthine oxidase, which ends up producing hydrogen peroxide and xanthine. And xanthine can also be produced from guanine, which undergoes a hydrolytic removal of its amino group right here to end up yielding the xanthine. And then the xanthine goes to this oxidation hydrolysis with xanthine oxidase, still producing hydrogen peroxide, and ultimately having uric acid, which gets deprotonate and have urate. But if you have overproduction of urate that ends up becoming a salt form with sodium salt, then that kind of crystallizes in your joints and actually cause inflammation. And that is people who suffer from gout is actually experiencing. And then finally, the catabolism of pyridines. And so the pyridines that you have can, in the pathways of degradation of pyridines generally lead to the production of ammonium, and then thus goes to the urea synthesis, so the urea cycle. So remember having that ammonium and then the carbon dioxide, which ends up being bicarbonate, and then goes through the urea cycle to end up creating urea that gets excreted out. But there's other degradations that can happen. So the thiamine can be degraded down to be succinyl coenzyme A, and then cytosine and uracil can also be broken down to end up creating acetyl coenzyme A, and that concludes the final part of the series, nucleotide biosynthesis.